PhD at the University of Utah, and he is currently a postdoc through the International NSF Postdoctoral Fellowship at the Universidad Nacional de San Luis, and he's jointly advised by Seth at Vanderbilt. And today, Kevin will be talking to us about friends for life, our friends for life, gut microbes that allow herbivores to consume toxic plants. So please welcome Kevin Kohl.
Thank you. I'd like to thank the organizing committee for selecting me. It's an honor to be here. I've really enjoyed the talk so far, and I'm looking forward to the rest of the day. And so the theme of this meeting is the role of microbiomes in ecology and evolution. And I'm personally particularly interested in the roles that microbes play in animals, ecology, and evolution. And my training is sort of in animal physiology, and I'm particularly interested in the processes of feeding and digestion. It's really through these processes that animals acquire the energy and nutrients that they need to survive and reproduce. And so feeding and nutrition are very important for the fitness of animals. Particularly, I've been studying mammalian herbivores and their interactions with gut microbes. Now, herbivory is a common and derived feeding strategy in mammals. It's common in the fact that about 40% of mammalian species are herbivorous. And by looking at the pictures here, we can see that this feeding strategy has evolved numerous times independently. Now, this might lead you to believe that herbivory is a relatively easy feeding strategy. However, this is not the case, as plants are extremely low in nitrogen or protein and high in indigestible materials such as fiber. And so herbivores host microbial communities to aid them in overcoming these nutritive challenges. Now, in addition to these nutritive challenges, plants also produce a wide array of plant secondary compounds that act to deter feeding by herbivores. And decades of research have investigated the roles that these chemicals play in determining foraging strategies of mammalian herbivores. Now, research in this area is important for a number of reasons. First of all, many herbivores can play very large roles in determining whole ecosystem structure by selecting different plant species to consume. And so the role that these chemicals play in altering that diet selection can then scale up to ecosystem structure. Additionally, many agricultural herbivores are often faced with these same plant toxins, especially in grazing systems. And so by understanding how wild herbivores cope with these toxins, we might gain better insights into agricultural practices. And so in 1974, renowned ecologists Freeland and Jansen published a seminal paper discussing strategies that mammalian herbivores use to overcome these toxic challenges. And they set up a number of hypotheses that have been worked on since this publication. However, one idea, that of interactions between gut microbes and dietary toxins, has received very little attention since it was proposed in this paper. And so today I'm going to present a series of experiments addressing this idea of interactions between plant toxins and gut microbes. I'll first walk through a background of the study system that I've been using to investigate these questions. And then I'll present two experiments testing hypotheses originally set up by Freeland and Jansen in their 1974 paper. First, testing whether toxins sculpt gut microbial community structures, and then also whether gut microbes actually facilitate the ingestion of toxins. And then I'll finish with an experiment looking at whether toxin tolerance can be transferred across species, which is really important for this idea of whether these microbes could be used in the agricultural industry. So I'll start with a little bit of background. I've been investigating these questions in wood rats, which are small herbivorous rodents in the genus Neotoma. This is a diverse genus, and it's really interesting because species tend to specialize on very toxic plants. And so today I'll focus on one species, the desert wood rat, and the toxic shrub that it consumes, creosote bush. So in the Mojave Desert of the southwestern US, some populations of the desert wood rat specialize on creosote bush, such that it makes up to about 75% of its daily diet. Now, creosote is a very toxic plant. It covers its leaves in a phenolic-rich resin that can make up to 20% of the dry matter of the plant. This resin contains over 300 compounds, primarily phenolics, although about 40% of the resin is made up of a single compound, nordihydroguaric acid, or NDGA. Now, this compound is known to cause kidney cysts and liver damage in lab rats, And remarkably, every single day, the desert wood rat consumes the lethal dose of this chemical as measured in lab mice. And so these guys are really remarkable at consuming plant toxins. But that's not the whole story. Just a little bit more of the natural history of these species. So this map shows the state of Utah, and the range of the desert wood rat is shown in orange. Now, creosote naturally invaded about 17,000 years ago from the south, just into the southern portion of the range of the desert wood rat, so shown in these red lines. And so it sets up this really interesting natural scenario where we have southern populations that have 17,000 years of ecological and evolutionary experience with creosote bush, which I'll call the experienced populations. And then we have these northern populations, which I'll call the naive populations, that lack this ecological and evolutionary experience. And interestingly, when we bring these populations into the lab, they exhibit differential tolerance to creosote toxins, such that the experienced population can consume about 25% more toxins, suggesting that there's some sort of adaptation in this group. Wood rats are also interesting from a microbial perspective because they have sort of a unique gut morphology. So this is a diagram of their gut. They maintain a large cecum where they perform their fiber fermentation. But they also have a semi-segmented stomach, sort of similar to that of a cow rumen. And so they maintain this foregut chamber that was actually hypothesized for over a century to house a dense microbial community. And indeed, we showed that this chamber does house a dense microbial community. So this graph, we have our different gut regions, and we used flow cytometry to measure the number of live cells per gram of contents. And we see that the foregut has a density of microbes on par with that of other well-known microbial chambers, such as the cecum or large intestine. Now, the residence time of this chamber is relatively short, and so it's unlikely that any extensive fiber fermentation is happening here. And so rather, we sort of hypothesize that this is a detoxification chamber in order to allow for these chemicals to be metabolized early on in the gut before they would be absorbed in the small intestine. Just one last piece of background. We were also curious whether our animals maintain their natural microbiota when we bring them into captivity. And we found that after 6 months in captivity, 64% of the microbial OTUs, or operational taxonomic units, which you can sort of think of as a bacterial species, are maintained. And so this is really important to know so that we're studying a relevant natural microbiota in our lab-based studies. So just as a little summary, wood rats really represent a tractable system in which we can begin to investigate these questions of interactions between gut microbes and dietary toxins. They house this dense foregut microbial community that we think is important for detoxification, and they maintain their natural microbiota for our lab-based studies. So I'll now present a series of experiments testing this hypothesis, originally set up by Freeland and Jansen, asking whether toxins sculpt gut microbial communities. And so to do this, I brought individuals from both the naive and the experienced populations into captivity and placed them on either a control diet of rabbit chow or a diet containing 2% of extracted creosote resin. We can extract the creosote resin from the plant and add it to our base diet of rabbit chow. And so any differences we see are solely due to the addition of toxins and not any nutritional differences between plant species. We fed animals these diets for a week, and then we collected contents of that foregut chamber and inventoried microbial communities by sequencing the 16S rRNA gene. And so with that, we get community profiles and relative abundances of different groups. And so here are data for the naive population fed either a control or a creosote diet. And we have the relative abundances of different bacterial phyla. And here we can see that when naive individuals are fed a creosote diet, the relative abundance of Firmicutes increases and the relative abundance of Bacteroidetes decreases. Now, interestingly, when we look at the experienced population, we see a different shift, such that when these animals are fed creosote, the abundance of Firmicutes decreases and the abundance of actinobacteria increases. And so these results show that indeed toxins do sculpt microbial community structure, but that these shifts are dependent on previous experience with the toxins. Now, these data only look at what types of bacteria are there, not necessarily what they're capable of doing. And so for that, we performed metagenomic sequencing, where we sequence all of the DNA in a mixed sample, and we can classify reads to particular functions. And so we did that for the experienced population, fed either the control or creosote diet. For the rest of my talk, I'll be switching back and forth a little bit between the two populations, and so I'll always have a figure corresponding to what population I'm discussing. So again, this is metagenomic sequencing from the experienced population. And I'll present the results in the form of a heat map. This is sort of a complex figure, but I'll walk you through it. So here, each row represents a functional category that those reads were assigned to, and the darkness of the box corresponds to how abundant that function is. And so here we can see that carbohydrate metabolism is a dominant function of the wood rat foregut microbiota, whereas sulfur metabolism is not as abundant. Each column represents an individual animal, and so we can look at that functional profile of an animal and compare it across our groups. So then we can look at these whole functional profiles and cluster animals according to similarities in their functional profiles. And we see that the control and the creosote-fed animals cluster distinctly. And the majority of this is driven by these top two categories, the stress response and the metabolism of aromatic compounds, which are overrepresented in our creosote-fed animals. So it does seem that toxins do alter microbial community function. Now, each of these functional categories is a relatively broad descriptive term. And so we can go within each of these terms to look at more specific functions or even specific genes. And so we did that for the metabolism of aromatic compounds and found that this differential abundance is primarily driven by a differential abundance in a single gene, aryl alcohol dehydrogenase. Now, this gene converts aromatic alcohols into aldehydes and plays a key role in the degradation of compounds such as toluene, biphenyl, and phenol. And if we look at the chemical structures of these compounds, it might not be so far off as to think that this gene could be playing a role in the degradation of NDGA, which is the most common compound in creosote resin. So as a summary for this section, it does seem that dietary toxins do alter microbial community structure. Taxonomic inventories showed that the foregut microbiota is responsive to toxins on both a short and long-term timescales, such that feeding toxins alters the community, but that shift is dependent on previous experience. And when we look at functional capabilities, toxins also sculpt those. We see an overrepresentation of genes associated with the metabolism of aromatic compounds, and especially this aryl alcohol dehydrogenase, which we're interested in following up on. All right. So even though we see these shifts in microbial community structure and function, that still doesn't necessarily tell us whether these microbes are important in allowing herbivores to consume toxic plants. And so we were still left wondering whether microbes facilitate the ingestion of toxins. And so we performed a number of experiments to test this question. We first used antibiotics to reduce the microbiota and looked for an effect on their toxin tolerance. So for this experiment, we collected individuals from the experienced population and brought them into captivity and placed them on a number of different treatments. They were fed either a control diet or a diet with 2% of extracted creosote resin, and a subset of animals within each of these diets were placed on antibiotics. We used neomycin, which is a broad-spectrum antibiotic that doesn't cross the gut lining very well, and so any differences we see should be solely due to reduction in the microbiota and not any systemic effects. We fed or maintained animals on these treatments for 2 weeks and monitored things like food intake and body mass. And we removed animals from the trial when they lost more than 10% of their original body mass. The animals actually prefer to starve themselves rather than poison themselves. And so this loss in body mass is a very nice metric in order to look at overall tolerance. And so the results are in the form of a Kaplan-Meier survival curve. Again, the animals didn't die. They were removed from the experiment. And so we have our days of the trial on the x-axis and the proportion of wood rats persisting in the trial on the y-axis. This black line represents animals that were not given any antibiotics and were fed either a control or a creosote diet. And we see that all animals persist in the trial for the full 2 weeks. This blue line represents animals that were given antibiotics and fed a control diet. And we see that a few animals drop out, but this line is not statistically different from the black line. But what's most striking is when we look at animals that were given antibiotics and fed the creosote diet. Here we see that all animals drop out within 2 weeks. And this p-value compares the red line to the blue line. And so these data really show that reduction of the microbiota significantly impairs these animals' ability to feed on creosote toxins. So we were then curious whether this toxin tolerance could be transferred into the naive individuals. So just as a reminder, we have this natural scenario where the experienced animals are able to consume more creosote toxins in the laboratory. And so essentially what we wanted to do was transplant the microbiota from the experienced population into individuals of the naive population and see whether we could confer that tolerance. And so we performed a fecal transplant, which are all the rage right now, especially for human illness. Some people will even call it repopulating the gut. But I use the term microbial transplant. It's a little nicer. So for this, we started with 2 groups of naive recipients. And for one group, we took other individuals also collected from the naive population, collected their feces, and mixed it into the diet of our recipients in order to create a control transplant group. For our other group, we collected individuals from the experienced population, collected their feces, and mixed it into the diet of our recipients in order to create an experimental transplant group. And so here, we essentially have 2 groups of animals where the hosts were collected from the same population, and so they should share similar liver detoxification machinery. And the only difference is the type of microbiota that we introduced into them. So then we conducted a feeding trial where we increased the concentration of creosote resin in the diet every 3 days. We monitored various physiological parameters such as food intake and body mass, and again measured overall tolerance by removing animals from the trial when they lost more than 10% of their original body mass. So when we look at percent change in body mass, so here we have days of the trial on the x-axis, and these boxes correspond to what was happening in the experiment at that time. So this is the percent creosote resin, and this is when the microbial transplant was occurring. And on the y-axis, we have percent change in body mass. So early on in the trial, we don't see any differences between the 2 groups. However, as they transition to a toxic diet, we see a significant difference such that the experimental transplant group was able to maintain higher body mass on these toxic diets. When we look at overall tolerance, again, removing animals from the trial when they lose more than 10% of their body mass. So here we have days of the trial and the percent of creosote resin. So both groups are retained early on in the trial, but as they're pushed to higher doses of creosote resin, we see a significant difference such that the experimental transplant group was able to consume higher doses of creosote resin and persist in the trial longer. So this really shows that the experienced microbiota increases host tolerance to toxins. And so as a summary for this section, gut microbes really do seem to allow herbivores to consume plant toxins. Reducing the microbiota significantly impairs their toxin tolerance, and the transplanting of the experienced microbiota increases the toxin tolerance of our naive individuals. So then we were curious whether this toxin tolerance could be transferred across species. And this is really important for this idea of using wild microbes in the agricultural industry. And the majority of this work was led by an undergraduate that I mentored, Ashley Stengel. And for Ashley's project, we were really interested in tannin-degrading bacteria. Tannins are a specific class of plant secondary compound, and they bind dietary protein and also digestive enzymes and inhibit them. And so they really limit the nutrient availability to an animal. And so they're of large concern in the agricultural industry because of this nutrient-limiting effect. And so there's been interest in isolating tannin-degrading bacteria that can be used in agricultural herbivores. And indeed, a number of tannin-degrading bacteria have been isolated. They were first isolated from the koala gut in the year 1990, and since then have been isolated from a number of wild and agricultural herbivores. However, even though we've been isolating these types of bacteria for 25 years, we still don't know whether they're functionally significant to their hosts. Or that is, we don't know whether hosting tannin-degrading bacteria allow mammals to consume higher doses of tannic acid. And so we designed a series of experiments to fill this knowledge gap. We first isolated tannin-degrading bacteria from wood rat feces. So this is just an agar plate that's been overlaid with tannic acid. The tannins bind the proteins in the agar, creating an opaque surface. And then we can culture wood rat feces on top of that. Microbes that are capable of dissociating protein-tannin complexes create small zones of clearance around their colonies. And then we measured tannase activity to be certain that they really were capable of degrading these compounds. And so using these techniques, we isolated 3 species of tannin-degrading bacteria from the wood rat gut, Bacillus subtilis, E. coli, and Enterococcus fecalis. And so then we performed a series of transplants in order to test whether these bacteria were important in allowing animals to consume tannins. So we transplanted them into lab rats. Now, wood rats and lab rats are in different taxonomic families and have been separated evolutionarily for about 25 million years. And so this does represent a substantial host switch for the animal, or for the microbes. For the first group, we collected whole wood rat feces and autoclaved them in order to sterilize them and mix them into the diet of our recipient lab rats in order to create a control group. For another group, we grew up our tannin-degrading isolates in an overnight culture and mixed them into the diet of our recipients in order to create an isolates group. And then for a third group, we collected whole wood rat feces and mixed it into the diet of our recipients in order to create a community transplant group. And so similar to my last transplant experiment, here we have 3 groups of animals where the hosts are probably genetically identical, and so they share similar liver detoxification machinery. It's just the type of microbes that have been introduced into them that differ. And so then we conducted a feeding trial where we increased the concentration of tannic acid in the diet and then held them on a 12% diet for 9 days. We monitored various physiological parameters such as food intake and body mass. And in this experiment, we also measured an indicator of liver damage. And then we measured overall tolerance by removing animals from the trial when they lost more than 10% of their original body mass. And so when we look at food intake, here we have our 3 groups and the cumulative tannic acid intake on the y-axis. And we see that both the isolates and community groups were able to consume about 12% more tannic acid in the beginning part of the experiment. And so this shows that hosting tannin-degrading bacteria do allow hosts to consume higher doses of tannins. Now, interestingly, despite this higher toxin intake, these groups actually showed lower indicators of liver damage. We measured alanine aminotransferase activity in the serum. This enzyme is primarily produced by liver cells, but as the liver cells rupture, the enzyme is dumped into the serum where its activity can then be measured. And so we see that at least the community group shows lower indicators of liver damage despite having a higher toxin intake. And then again, we measured overall tolerance by removing animals from the trial when they lost more than 10% of their original body mass. So here we have days of the trial and the percent of animals persisting in the experiment. And our control animals all dropped out within 21 days. But if we look at animals that were given tannin-degrading bacteria, either as isolates or as a full community, these animals persist in the trial significantly longer, really showing that these microbes are important for or increase performance on tannin-rich diets. So we then wondered whether this function was retained even in the lack of dietary toxins. And this idea is really important for the idea of using these microbes in the agricultural industry where repeated inoculation might not be feasible. And so this graph is the graph that I just showed you on the previous slide. And we then allowed the animals to enter a 1-month recovery period during which we didn't feed them any tannins, just basic rat chow. And we then reconducted the feeding trial on tannic acid and basically found the same results. suggesting that these microbes are retained in the gut and their function is also retained. So as a summary for this, it really does seem that tannin-degrading bacteria do allow hosts to consume higher doses of tannins. This is the first functional test of these bacteria, even though we've been isolating these types of bacteria for 25 years. And I think it really shows that microbes from wild herbivores might be of use in the agricultural industry in order for conferring toxin tolerance to naive species. And so if we bring this back to Freeland and Jansen, who originally proposed some of these hypotheses in 1974, this work really represents some of the first progress made in this area. And it does seem that gut microbes do facilitate the ingestion of dietary toxins. And I think we can take this out to herbivores as a whole. I think it's likely that microbes drive the foraging strategies of other mammalian herbivores. My studies were very rodent-focused, but I think it'd be interesting to look at other mammalian groups and also other chemical classes of defenses. And I think it'd be really interesting if scenarios exist where a certain gut microbe changes the foraging strategy of a mammalian herbivore that then scales up to whole ecosystem structure by allowing that mammal to consume a different plant. I think these microbes might also be really important for rapid adaptation to changing environments. With climate change, the range distributions of plants are expected to change. And so herbivores might be faced with very novel classes of plant toxins. And so it might be easier to acquire a new microbiota than to evolve a new liver detoxification enzyme. And finally, I think wild herbivores might be a novel source of microbes that could be used in the agricultural industry. And this is really highlighted by the tannin-degrading bacteria experiment. And so if we take this out to sort of feeding and digestion as a whole, I think we're really at the forefront of a new area of research in understanding the roles that microbes might play in feeding and digestion, and thus the ecology and evolution of animals. And so I think in the next few years, we're really going to uncover some unexpected and exciting roles that microbes can play. So with that, I'd like to thank a number of collaborators and funding sources. Again, I'd like to thank the organizers for inviting me, and all of you for attending, and I'll take any questions. Mm-hmm.
But then you were able to retain that effect of the fecal transplant in the absence of the tannins.
Mm-hmm.
How do you think this happens outside, and how do you reconcile those two things? It seems that diet affects microbial composition, but also you can retain some function over a long time without that diet.
Right. So my first experiment showed that that toxins do sculpt microbial community structure, but I was showing relative abundances. The addition of toxins actually didn't change the representation in that it didn't cause the addition or loss of any microbial species. And so I think it's through that that function is then retained. And so in the rat experiment, we were curious, do these tannin-degrading bacteria require tannic acid to survive in the gut? And it seems like they don't. So they, as they're taken off tannic acid, their gut community likely changes, but those tannin-degrading bacteria are retained at presumably low levels and then allowed to re-increase.
Do you think over that time period they were able to re-increase? I mean, you do need the similar relative abundance presumably to have a similar effect on survival, or no?
In the tannic acid study?
Yeah.
We unfortunately didn't collect samples to compare on and off tannic acid, so I don't have that answer. But other people sort of before 16S sequencing have shown that tannins do change the microbial community structure. But I think it's an issue of structure versus representation, or like who's there versus how much of them are there. Yeah?
So do you have a sense of whether the tannin-degrading bacteria are deleterious in the absence of selective pressure? So just on a non-tannic acid-containing diet, will the rats do as well, or do the mice do? You know, is there a fitness cost?
Right.
To bacteria?
That's a good question. And maybe our rats probably aren't the best system to study that in captivity. It could be in the wild that they do have some deleterious effect when there are more selective pressures than in the laboratory. But at least, I mean, when they were taken off the tannic acid, all the rats increased food intake, regained body mass, and so they were presumably doing well. But yeah, that's a good point. Yeah.
Do you know if there's any anecdotal reports or evidence of herbivores intentionally inoculating themselves to other species?
Yeah, one question that I often get is where do our wood rats get their microbial communities? And wood rats are really interesting animals. They build these huge midden or nest structures, and they go out and collect coyote feces and cow patties and things like that. And they also perform coprophagy, or the ingestion of their own feces, and they will eat stored feces. And there are instances where species will cohabitate in a den. In terms of purposefully inoculating themselves, iguanas are herbivorous, and they're known to consume the feces of adults. The juveniles are. Some herbivorous birds will do the same thing. They'll consume the feces of adults, presumably to transfer that microbiota, things like that. Yeah?
Kind of along the lines of test talk, do you see any evidence for the naive and experienced populations maybe having genetic divergence because of that difference in diet, and that might be related to their different microbiome?
In terms of genetics per se, we don't know. We're actually working on the wood rat genome. We have my PI has done a lot of work on liver gene expression. And similar to the microbiome where we see these different shifts on a creosote diet, the liver responds very differently to creosote toxins, the naive versus the experienced population. Different genes are turned on, and so we think that's probably through evolution of genes associated with gene expression, but it could also be driven by the microbiome up through host gene expression. And so we're doing some experiments to look at that as well.
So first, before my real question, to clarify, were the results from those who received the transplant the same as those in the experienced population that already had, or was it like less strong?
That is, if I could go back in time and redo something, I would have added an experiment or a regular experienced animal to that group. I wish I would have. Based on comparing experiments, they didn't do quite as well as a normal experienced animal would have done, but I didn't run those animals in parallel.
So I know that at least, I don't know about most herbivores, but I know that koalas, they get most of their gut bacteria by eating the feces of their mothers when they're babies.
Mm-hmm.
So do you think that young individuals from a naive population, if they receive this gut bacteria from the adult of the experienced population, would they do as successfully as the experienced population? Would it differ depending on the age they received the gut bacteria?
That's a really good question, and something we would love to do. We've had a number of undergraduates try to breed wood rats, and we just haven't been successful. But that would be really interesting to sort of disentangle how much the microbiome is doing versus maybe the liver detoxification machinery. I will say that we sometimes catch pregnant females, and they give birth in captivity. We've compared mother and offspring, and they basically have the same microbial community. So there's very strong maternal transmission, but we haven't been able to do that breeding experiment.
Thank you so much.
Mm-hmm.